So the second line of evidence for gene flow that we looked at was based on haplotype structure. And the idea is the following. We look for sites in the genome not using the Neanderthal sequence at all. We look for places in the genome where there are deeply diverged haplotypes, very deep genealogies, places where there's some people have lots of SNPs that are not seen in anybody else, that are only seen in non-Africans. This is very unusual because, of course, we know Africans have much more diversity than other populations. So finding a place where non-Africans have more diversity is very weird and might be evidence that that bit of the genome has had an influx from an archaic population. People have always been interested in this. Here's a list of 5 papers that have found candidate such regions. The first one was Damien Labuta's paper on the dystrophin exon. More recently, Jeff Wall and colleagues looked at a large number of loci and found that there were more of these than you would expect based on most demographic models, suggesting maybe archaic flow in non-African populations. But without a Neanderthal genome, you can't really tell whether these are from Neanderthal or not. So what we did here is an exciting thing that happened in this project is Rasmus Nielsen and Weiwei Zhai were doing a study looking for such regions without being part of a consortium at all. So they approached us and they said, we have this list of regions of the genome that we found that are candidates for ancient gene flow. Could we look at the Neanderthal genome and see what the Neanderthal has? So this is a great experiment because it wasn't done by us. It was done by outside person without any access to the Neanderthal genome. And so we looked them up. So they developed a way of going through a genome-wide data set. They started with HapMap. We moved eventually to Perlegen, but got the same result. To find a list of such regions, they found 12 regions where they were able to identify tag SNPs that separated the variant that separated all the cosmopolitan group, which included Africans and non-Africans, from just the non-African specific deep group. And then we lifted the veil on the Neanderthal sequence and said, what did the Neanderthal have? And so here are the 12 regions, and they are listed here. Their average frequency is not so high. These are in terms of the variant seen only in non-Africans. It averages about 13%. And you can ask, did the tag SNPs tend to match that for the non-African specific type, the very ancient type? Do they tend to match the Neanderthal or the non-Neanderthal type? And what you see is an amazing thing. 10 out of these 12 regions have a vast excess of matching the Neanderthal. So these are variants that are only 13% frequency. They're only 13% frequency, and in Africans they're 0% frequency, or very close to 0% frequency. So the expectation is that in the common ancestral population of Neanderthals and modern humans, in the absence of gene flow or some kind of population interaction, they wouldn't be much more than 13% frequency. Maybe in the most extreme cases that you can imagine, maybe 50% frequency. Never more than that. And yet we see 10 out of 12 matches. And we think that that's kind of a crazy result that we can't sort of find any explanation of. An important thing to realize is this can't be due to contamination. Imagine all our data was contaminated. Then what you would see is maybe a 13% matching to Neanderthal, because we're actually just matching to the variant that's only 13% frequency in Europeans. But instead it's 10 out of 12. So if it was all contamination, you wouldn't get a higher matching rate than the frequency of these variants unique to non-Africans, but it's 10 out of 12. So that's kind of a result that can't be explained by contamination. And importantly, it can't be explained by gene flow into Neanderthals, because these regions are discovered in modern humans without access to Neanderthals, so they're prioritized in modern humans. So it suggests that this must be due to gene flow into modern humans. The third line of evidence is that we look at human sequences where we actually have DNA sequence data, not just genotype haplotype data, and we look for regions with low divergence to Neanderthal. If there's no gene flow, you expect that the time to the common ancestor of, say, a section of the human genome reference sequence to the Neanderthal will be no more recent than what we estimate is the time to the split time, about 350,000 years. And if you take a second human, like the Venter genome, and you look at the time to the common ancestor with the human reference sequence, it will be just typical because the common ancestor will have lived here, so it won't be particularly high or low. If you look at gene flow scenario, what you're looking at is a case where the human reference sequence shares a common ancestor with Neanderthal much more recently, and then followed a lineage living in the Neanderthal population. So in that case, you expect the time to the common ancestor to be typical of Neanderthals, so not 500,000 years, which is what we think it is per 2 modern humans, but more like 800,000 years. So we tested. So here's African sequence from the human reference genome. The human reference genome we estimate is about 1/3 African in origin, and we know where these segments are. So we restricted to these segments from RPCI 11, who's an African-American individual who comprises most of the reference genome. And We looked at the Neanderthal divergence to the reference genome versus the Venter divergence to the reference genome, and you see a nice linear correlation, no bumps. But when you look at the European segments, you see this amazing rise. So when the divergence to Neanderthal in the reference sequence becomes very low, you might expect, why should it be high to Venter? But it is. It's extraordinarily high. It's above the genome-wide average. It's almost as high as that we estimate to Neanderthals. So this is a third line of evidence. Again, it can't be explained by gene flow in the other direction, because that would cause this blue curve to dip, not to rise. If you look in the regions of very low divergence between the Neanderthal and the human reference sequence, but high divergence between Venter and the reference sequence, you see that overall there's about the same number of segments of the genome backs in the European and African segments, but in this top corner, I think it's 29 out of 31 are of European segments. So there's this huge enrichment, meaning that in these segments we can label these as really likely to be Neanderthal-derived segments with high probability. So if gene flow explains the data, you can actually ask what proportion of our genetic ancestry comes from Neanderthal under this hypothesis of gene flow. And the way we did this is a very simple way. Remember we did this experiment, the first line of evidence for gene flow, where we asked at a site that's different between an African and a French individual, what's the rate at which they match to Neanderthal? So we now can add a second Neanderthal and say how much of the way to a second Neanderthal is a French person. And when you do this, they are about 1% of the way to a Neanderthal, which turns into an estimate of at least 1% gene flow. We also did a modeling analysis and got numbers 1 to 4%. Our current estimate's about 2.5%. So a caveat is that this analysis is not distinguished between gene flow from an ancient structure scenario that could potentially explain the data. So here is the model of gene flow. An important thing to point out is that if you remember, the gene flow signal affected the Chinese sample and the Papuan sample and the European equally, as far as we could tell from this data. So that means that you're not seeing evidence specifically of an interaction in Europe. There's no evidence, even though that's where the archaeology suggests the overlap. The most parsimonious explanation is you're seeing evidence of an interaction here in the Levant before the diversification of non-African populations during the after the out of Africa dispersal. It doesn't mean that gene flow in Europe didn't occur, just that we don't have power to detect it. But the other explanation is ancient structure. So if you imagine that, say, 350,000 years ago, the human population was substructured into 3 groups, here 1, 2, and 3, one in the Near East, one in Northeast Africa, and one more Southern Africa, exchanging genes, but quite differentiated like human populations today. And then what happened is population 1 went off and became Neanderthals a few 100,000 years later, and populations 2 and 3 continued to exchange genes and then evolved into modern humans, but population 2 retained a little bit of extra proximity to Neanderthals because it started out that way, not complete homogenization. Then during the out of Africa expansion, which potentially was from a Northeast population, it would have retained a little bit of extra proximity to Neanderthals. So is that explaining our data? So what's exciting is that we now have sequence that Svante Pääbo talked about at the Cold Spring Harbor meeting in May, from a 40,000-year-old or maybe 50,000-year-old ancient bone from Denisova cave in Russia, which has an amazing mitochondrial DNA, whereas the time to the common ancestor of human and Neanderthal mitochondrial DNAs is about 500,000. This is about 1 million years. It's like another mitochondrial lineage we've never seen before. And we've now carried out genome scale sequencing. We have actually more data and better quality data than the Neanderthal, about 1.9x coverage. And when you build a cladogram, and the branches are proportional to the time to the common ancestor, Denisova is a sister group to the Neanderthals. Here's 4 Neanderthal individuals. And this is different from the mitochondrial DNA, where Denisova is an outgroup to modern humans and to Neanderthals, but the nuclear genome tells a different story, where they're sister groups, but distant sister groups, slightly more diverged than the most distantly related human populations. So this is the tree that we think may apply to the data, and the question is, which of these 2 stories applies, ancient structure or gene flow? Well, ancient structure makes a prediction. If they're really sister groups, perfect sister groups, then the ancient structure in Africa should leave the same effect on Denisova and Neanderthal. That is, the structuring that existed there would have existed in the ancestral population already and would have been propagated equally to them. So you'd predict the same relationship of exactly Denisova and Neanderthal to modern humans. And if it was gene flow instead, you would expect that Neanderthals would be particularly closer. But what you see is that Neanderthal is closer. So if you measure this asymmetry statistic, the first line of evidence, it's 5.1% excess matching to French and Neanderthals, but only 1.8% in Denisova. And that's a significant excess, but these are very different numbers. And out of these 10 out of 12 regions that Weiwei and Rasmus found, matching Neanderthal, a subset of those, 6 out of 12, are matching Denisova. So that tells you that this is less likely to be true and that the gene flow is more likely to be true. So the conclusions are that Neanderthals are more closely genetically related to non-Africans than to Africans, that it was likely but not certainly yet due to gene flow from Neanderthals into the ancestors of all non-Africans at that point. That the 1 to 4% ancestry that we estimate exists outside of Africa, it seems homogeneous outside of Africa as far as we can measure so far, but each non-African's Neanderthal ancestry is different, so where you get a Neanderthal allele may be different where someone else gets a Neanderthal allele. And a medical implication is that there's been this vast influx of novel SNPs that have come into modern humans around 50,000 years ago. We estimate that maybe 1 million new SNPs came in into a population that already had only about 10 million, and so they got injected in at a high frequency, 2% or so, up to 2%, where they would have potentially had an important medical impact, where selection could have acted on them. Normally, new mutations occur very rarely. Selection doesn't work very effectively on them. So this is potentially very interesting to think about what these alleles will do. So I finally wanted to thank the people who led this project, to Svante Pääbo, who's been working on this project for decades, and to Ed Green, who's the postdoc who led this work and the bioinformatic work and really interesting work on natural selection that I didn't have time to cover. And I don't know how much time I have for questions.
We have time for quick questions.